What is up? What's up, Arjun? Hola, hola. I feel like asparagus stuck in my teeth. I'm trying to eat. I got my time. I was crunched. I was crunched for time. So I'm eating right now. I got asparagus stuck in my teeth. What up, Arthur? I don't think I know you, Arthur. I don't recognize your name. What's up, Charvy? How you doing? Oh. Mr. Chang, as usual. How goes it? I did not go to the Sunnyvale Art and Wine Festival. The Art and Wine festivals, they're always the same. When you've been to one, you've been to them all. That's my opinion on them. You're a learner? All right. Okay. What's up, Meenakshi? How are you? Do I have any plans for the summer? I do indeed. Got big plans. I'm going to Eastern Europe. Well, actually, it's technically the Balkans. From what I've learned, is this area is called the Balkans for 6 weeks. I'm gonna start in Montenegro, and then I'm gonna go to Croatia, and then Slovenia, and then Hungary, Austria, Budapest. Budapest is in Hungary. And then, I don't know, maybe in Amsterdam, something like that. Be 6-week little backpacking trip. Gonna leave on middle of June, come back the end of July. It's gonna be a blast. It's a long trip, but it's kind of like what I like to do with my summers. I like to do these long backpacking trips. Started getting into that a few years ago. I missed you then. Okay, I would have seen you. I would have seen you there if I had come. Yeah, Croatia's supposed to be beautiful. My only problem with Croatia is I hope that it's not overcrowded because of all the Game of Thrones fans. Because they filmed a bunch of Game of Thrones scenes there. So people go there for like the tourism part. Meenakshi, do you need to know anything besides DNA replication and protein synthesis for the genetics unit? You should know a little bit of some of the biotech things that we covered. So remember things like stem cells. You should know things like recombinant DNA, technology, gene therapy, those kinds of things. Just kind of refresh on the basics of those. Just be able to identify what they are. Sanvi, what's up, Sanvi? What exactly in evolution is going to be on the test? Everything, the whole thing. You got to understand what evolution by natural selection is. You should be able to explain Darwin's theory. You should be able to talk a little bit about population genetics, sort of how allele frequencies change over time. You should kind of know some examples of evolution, like directional selection versus disruptive versus stabilizing selection. You should know, what else? You know, like some of the other things we kind of talked about, like homologous structures versus analogous structures, vestigial structures. Pretty much, there's really not much in there that we're not going to be on the final. To be be honest. Evolution is going to be pretty much covered. The Maldives not going to be there in 10 years? What? Because of like rising sea levels? I don't know. It's good. I'm not sure. I haven't heard that one, Shalini. I wouldn't be surprised though. But I'm not going to the Maldives. They look beautiful though. Look at the water. What the heck? Look at that. That's a little bit. It's a little bit much for me. A little bit much. Let's see. What do we got? Questions. Oh yeah, Arjun. That's where one of my first cities is Dubrovnik. It's where yeah, King's Landing is filmed in Dubrovnik. And then I think I'm also gonna go to Split, which is where Bravos is supposed to be filmed. So I'm gonna see quite a bit of it. Let's see. Kaushal, will meiosis and mitosis be on the test? No, that was first semester. You do not even know the specifics of that. Mahit, are the biotechs? What are the biotechs that we learned? We learned. I'm gonna pull it up real quick. Pull it up. Pull it up. Pull it up. Just to remind me. Of what just so I don't miss any. Should be on your sheet though, to be honest. It should be on the unit guide, the resource guide. Yeah, genetic testing, like genomics. Remember we did the presentations on it? Show you here, right here. These topics. Can you see that? Genomics, genetic testing, assisted reproductive technology, genetically modified organisms, stem cells, cloning, gene therapy, eugenics. Those are the topics. You guys want to do Kahoot again? The same one we did in class? It's not going to be boring because we'll just be repeating questions. Although we didn't finish it. We only got through like 30 questions. Shalini, are the questions? They're pretty similar. I mean, think about it this way. We cover the same material. So the questions are going to cover the same content. They're going to be a little bit different, but similar in a way. I can't say exactly how close they are, though. There's going to be a lot of overlap, though. Danush, what's up? What are the functions of DNA polymerase? So DNA polymerase does it synthesizes the new strand of DNA during DNA replication, and it also checks for errors, for mutations that have occurred and fixes them after replication has happened. I guess we could just do the Kahoot. We can throw the Kahoot up because there's no point in not doing it. We just have it running. I can still answer other people's questions in the chat too, so let's do that. Just gonna finish my food real quick. finish my dinner and get a chance to eat. I just got home from jiu-jitsu at like 7:45. I hopped in the shower. Gotta eat. What are the common mistakes on the final? That's a good question. Common mistakes on the final. I don't know if I have an answer to that question, actually, Shalini. The common mistakes on the final. It's almost like there's not really any, like, particularly like common mistakes. Spring final review. Yeah, I wish I could help you with that question more, Shalini, but I don't really know of anything in particular. It's like a common mistake. You like your group strategy? That's good. That is a good thing. Okay. Let's do a classic, one device. Yeah. What happened? So last time, what was the issue? You can't do the two-step because it's too delayed. Let's do a classic. Danush, does DNA polymerase also take out the RNA primer? Not that I'm aware of, but I didn't really talk about that in my class. I didn't get into that level of detail with the RNA primer thing. Mahed, I definitely can explain the evolution of skin color. I'll be right back though. I need to get a tissue. My nose is kind of running. Be right back. Not that many people, huh? All right, so let's just do it. I was going to answer Mahit's question. Can I explain the evolution of skin color? I sure can. So the way you can think about the evolution of skin color is basically this. What is the function of having darker skin, right? So in the sense of having darker skin, it helps block UV light. So where is there more UV light? There's more UV light at the equator. So you're going to have a ton of UV light at the equator, and you're going to have less as you go north or south away from the equator towards the poles. Okay. So the problem is UV light damages folate, which is a B vitamin. It destroys B vitamin. Oh, am I covering up the page? Yeah. I'm moving myself out of the way. There we go. Sorry for those of you who got that one wrong. You just had to make a guess. But as you go north or south from the equator, there becomes less UV light, so you don't have as much damage to your folate. So you don't need to produce as much melanin, which is the pigment in your skin that makes it more dark and blocks UV light. So the idea would be that your skin would get lighter as you go towards the poles, because you don't need as much melanin. The melanin is not being selected for. That production is not being selected for. And in fact, lighter skin is being selected for because the sunlight also triggers the production of vitamin D, right? And so if there's less sunlight or UV light available at the poles, you don't want melanin blocking the little UV light that there is, because it's going to stop the production of vitamin D in the body, which could give you a vitamin D deficiency. So actually, vitamin D is selected for at the poles. Light skin is selected for at the poles because of vitamin D deficiency. Dark skin is selected for at the equators because of folate deficiency. Make sense? Yeah, anyone can join mid-game. Look at the key. The pin is down here in the bottom corner. Just use that pin to get in on the action. Only one person guessed right, and they're using my gamer tag. Oh, I'm still kind of blocking stuff, aren't I? How am I going to get out of the way? Maybe I go over here. No, I just gotta make myself tiny. Good job, good job. Danush, what does the enzyme ligase do? Sorry, I didn't get that question earlier. So basically, ligase is an enzyme that actually connects, kind of anneals or seals together the individual pieces, the chunks of DNA that were created on the lagging strand of RNA, of DNA synthesis. I did not get into that level of detail in my class. And so if you did in your class, you just need to know that the ligase is what connects the pieces. So on the lagging strand, when DNA replicates, right, one strand is replicated continuously in a nice smooth strand. It's easy. The other strand, it does it in chunks. Like the polymerase goes, makes a chunk, comes back, makes another chunk, comes back, makes another chunk. Those chunks need to be connected, and what connects them is ligase. Okay, most of you got this one wrong. Which of the following is not a function of the respiratory system? Remember, the respiratory system, its main job is to essentially take in oxygen but not deliver oxygen. So the delivery part, that is the circulatory system. Meenakshi, there's 100 questions. It's 100 multiple choice questions. All right, so this one is, think about it this way. If you have these newts that are in caves, right, they have eye spots on top of their head. The eye spots stay there even though they have no function because there's no selection pressure against them, right? That's the only reason why they stay. And so it's like a vestigial structure. Okay, so half you got it right, half you got it wrong. Which of the following is not correct about proteins? If you have too much protein, it actually puts pressure on the kidney because your kidney's job is to filter out urea, and urea is the waste product that's produced that contains nitrogen, and that is produced when kidneys are broken down in the body. All the other ones here are true. Right? A complete protein would be something that contains all of the amino acids. An essential amino acid is something that cannot be produced by the body, so it's essential in our diet. And then excess protein is consumed from foods with saturated fats. That's true because, like, most foods that have a lot of saturated fats are animal products, right? So you're talking about meats, red meats specifically. Okay, looks like my blood sugar is low. The three of you guys that picked the blue option, glucagon causes the pancreas to release glycogen. No, glucagon causes the pancreas to release glucose. Remember, just to recap blood sugar regulation, so the pancreas produces two hormones, insulin and glucagon. Glucagon is the hormone that's produced when your blood sugar gets too low, right? So let's say it's been a while since you've eaten a meal, and your blood sugar is going down and down and down and down. What's going to happen is your pancreas is going to sense that your blood sugar is low, and it's going to release this hormone called glucagon, which will go to your liver, and your liver will release glucose. We'll take the glycogen that's stored up in there and release and break it into glucose, putting it into the blood, bringing your blood sugar back up. If your blood sugar is too high, like you just ate a meal, and now your blood sugar is kind of spiked, the pancreas will release insulin, which will tell your muscle cells to take in glucose, and also tell your liver to store glucose in the form of glycogen. So they kind of work in opposite ways. Danush, what is rRNA? It is on the study sheet. rRNA? Yeah, that's ribosomal RNA. Yeah, Kisham. That is correct. It's the RNA that the ribosome is made of. So just as far as, at least for my class, treat the ribosome as the place. Let's treat rRNA and the ribosome kind of similar. Because you remember you have mRNA, tRNA, and rRNA, 3 different types of RNA that are used in protein synthesis. Wow, I feel equally across the board here. Which is following is not true. Emphysema equals inflammation of the bronchi. That one's not true because remember, emphysema is when you have the lung, the tissue, the sponginess of the lung is decreased. So like when you inhale and exhale, the actual alveoli can't expand and contract, right? So they're not. So when imagine like you're trying to inhale, but the volume of your chest cavity doesn't. The alveoli don't expand to increase their volume, so the air comes in. So it's almost like they lose their elasticity, and it makes it breathing very difficult. So that is not true. The rest of these are true. Ooh, is this a sex ed question? Easy peasy. Good job. You guys know what consent is. That's good. Get myself off that. Seems like a little bit more reasonable spot. Good. So either of those are good options. So if you look at this kind of situation, just to review what this is, what we're seeing here is atherosclerosis. So this is not a good situation if this is your blood vessel. If this is like one of your arteries, let's say this is your coronary artery, or that brings blood to your heart, or let's say this is like your carotid artery that brings blood to your brain. and you have a buildup of plaque in the wall of the artery that's expanding like this over time. Remember, this takes decades to accumulate. This doesn't happen overnight. This takes a really long time. But this gets bigger and bigger and bigger, and eventually it can impinge and impede the flow of blood, right, slowing it down. And worse off, what can happen is it can rupture, which is what we see in this last picture here. So this plaque has ruptured, and what you're going to see happen is these blood cells now clot on here, and what you develop is a clot where that rupture has happened. Now it's completely blocked, right? And so what you need to do is you need to break this clot apart if you're going to try and fix this. So the two options you can do to fix this are, one is a bypass surgery, where you basically take like an artery from, you know, another part of the person's body, usually their leg, and you would stitch it on, and you'd just go around the block. You'd make like a bypass, basically like a detour, so the blood can flow around it through a different artery. The other option in this case would be an angioplasty and a stent insertion. So what that means is an angioplasty you'd come in and you'd kind of like almost like try and remove some of the plaque, and then you'd insert like a metal case, a metal tube into the artery that pops open, and it props it open so that more blood can flow through. That's a stent. Yeah, TPA is a drug. It's a chemical that will break apart clots. So you could use TPA in this situation theoretically, but the problem is that it's not just the clot that's the issue here. It's this whole plaque, and TPA doesn't do anything for the plaque. It'll help break up the clot part, but it's not remedying the whole situation. And it's used for ischemic strokes. That is true, Danush. They sometimes use TPA for heart attacks as well, if they suspect that if they get to the person in time. The problem is TPA doesn't work after a certain period of time, because once the clot is too formed and too solid, the TPA molecule doesn't really work. But if you can get to it early, you can use it. What happens if plaque moves through your bloodstream? So, Charvy, plaque is not going to move through your bloodstream. That plaque is going to be stuck right here. What happens if it ruptures, it'll form a clot, and then that clot could break off and travel along your arteries. So you're not going to see the plaque really move, but you're going to see if a plaque ruptures and creates a clot, the clot can move. And then that clot could get lodged, you know, in your brain and cause a stroke, or in your heart and cause a heart attack. Nice, Shalini. You just nailed. You just explained it exactly the way I explained it. Good job. I'm gonna get some more water, so I'll be right back. Nice, good job, guys. Just remember that whoever you were that picked the blue one, remember each row here is not one nucleotide, it's actually two, right? So the orange, the phosphate group, which is the orange P, the pink hexagon in this case, or no, pentagon, excuse me. The pink pentagon is the sugar, and the blue structure is the nitrogen base. And those three things make up one nucleotide. So this is one nucleotide, the other side has another one, so there's 10 total. Okay, most of you got it right. RNA polymerase is not for DNA replication, right? That would be for protein synthesis. Pepsin, that's involved in digestion of proteins in the stomach, not involved in DNA replication. This one is kind of a tricky one. I don't know if I'm the biggest fan of the image that I selected for this, now that I think about it, because it can kind of be confusing. So the reason why it's homologous is because it has the same bone structure, like the same bone, same types of bones, right? They all have a humerus, they all have a radius, they all have the finger bones and thumb. It's all there, right? And so there's the same structure. The problem is it's also has, these are all wings, so that it all has the same function too. So it's kind of confusing, but these are definitely homologous. Shalini, what do you mean, am I going to do the water mixing thing for the pig practical? The water mixing thing? I don't know what that is. I'm not sure what you mean by that. Analogous would have been like if they were all wings, but they didn't have the same bone structure. So like they have the same function, they're all used for flying, but the underlying structure that makes them, like how they're created, it would be different. So like if you look at the wing of a bird and the wing of like a fly is an insect with a wing that's made of, you know, like a very, like, actually I don't even know what a fly's wing is made of. But it's not made of bone, right? There's no bones in a fly's wing. So the structure is very different, but they have the same function. So they're analogous. Yeah, that's why I didn't really like the picture, to be honest, Keshav, because it was kind of confusing because it was analogous structures, or they were all wings for flying, but they had homologous structures. So it was kind of a, it's still homologous because homologous structures can all have the same function. It's possible. So it's a little bit of a confusing question. Oh, you're talking about the colored water. Colored water. That's what you meant by that. Well, right now I'm just drinking some carbonated water from my soda stream. I make my own carbonated water. I just feel like carbonated water right now, just a little bit for some reason. Good job, guys. Easy. What's up, Aditya? You didn't miss too much, to be honest. You can just join in on a Kahoot. I answered a few questions in the chat, but that's about it. Three possible answers for this one. remember, reproductively isolated, right? That means that you have two populations. They're isolated. That means they do not mate with each other. So everyone over here just mates with each other. Everyone over here just mates with each other, and they never mate with the two groups never come together. So that means all of the genes that make up the individual groups are no longer mixing. So the alleles don't mix. Yeah, there's not that many people. Well, Aditya, there might have been more people that signed up to play, but then didn't finish. There's only like 8 people are answering at a time. This one's too easy. It's an easy question. It's a gimme. It's a gimme question. Shalini, do I remember you? Maybe. See, the problem is I'm much better at remembering faces than names. So I have to see you. But your name does sound familiar. Who do you have? Who's your bio teacher? Nice. We have learner. Man, I think I remember you from. Have you been on the stream, my stream, office hours stream before? Your name sounds familiar, but I'm not placing the name in the base. That's the problem right now. The reason why small population is. So just quickly hit this one. Right, if you have a very tiny population, there's just not that many individuals, so there's not a lot of differences. So that becomes an issue with small populations. The selection pressures are still there. That doesn't change whether it's a small or large population, right? The environment is the environment. Time doesn't really, it doesn't matter how many individuals are there. Time is time, right? The only thing that really changes is the variation as you get smaller.
Prokaryotic cells, translation is initiated by formation of an initiation complex consisting of the 30S ribosomal subunit.
Danush, what are histones, genes, and alleles?
50S ribosomal subunit then joins the complex.
Well, those are all different things. Genes. Well, let's start with histones. Histones. Nice, good job, guys. Histones just are the proteins that DNA wraps around to form tightly packed structures called chromosomes. So a histone is a protein that DNA wraps around in order to form a chromosome. A gene is a section of DNA that contains the instructions for making a certain protein. So it's like the instructions to make the protein. And then an allele is a version of a gene. So if I have a gene for eye color that determines the color of my eye, I can have the blue version, I can have the brown version. Those are the alleles. Oh, Shalini, you go to archery club. Okay. Yes, I'm pretty sure now I'm... Yeah. Now I got it. I got it. I got it. I got you. Yeah. Now I can put it... Now I pieced it together. That was the key piece of information. 3 options for this one. Those of you guys that put the green option, you probably didn't keep in mind this says veins. Atherosclerosis does not occur in veins because the pressure is not high enough to cause damage. If this said once the lining of the artery is damaged, then that would be a true statement. Too easy. Too easy. Some of these are a little too easy. A little too much. A little too much. Yeah, Charlie, that is fácil dinero. Shalini, yeah, the copper one doesn't use hormones. But usually, the most common ones that people use do contain hormones, from my understanding. Alright, not everyone got this one. Which is not a reason why bacteria are evolving resistance so quickly. So antibiotics have become less potent over time? No. The antibiotics are the antibiotics. Their potency hasn't changed, right? That's why that's not the reason. So those of you that put overuse in sanitizing products, that is true, right? Overuse, short generation times is true. The quicker, so an organism can evolve faster if it reproduces quicker, right? So if generation time is really important for the speed of evolution. That's why humans evolve much slower than bacteria, because we reproduce every, like, I don't know, 20 years, let's say. Bacteria is like they reproduce every 20 hours. So they're going to evolve way faster. Nice. Remember protein? Whoever you were that answered protein, that's going in the stomach. Protein starts in the stomach. What's up, Stefan? Is it Stefan? Steven? Stefan? Stefan? I never know Which pronunciation for that name? Lamarck, good. Can I list out where each biomolecule is digested? Yes. So, carbohydrates initially can be digested in the mouth. It's the first place where they can be digested. But they're also digested in the small intestine. So, the small intestine digests every biomolecule. Lipids, carbohydrates, proteins, and nucleic acids, I guess, but that's not really something I have to think about when we think about digestion. We mainly think about proteins, carbs, and fats. Those all are digested in the small intestine. The stomach can do protein, and the mouth can do carbs. So those of you that answered this one, which of the following is not true? So this is true. The pancreas is considered a digestive organ because it does secrete digestive enzymes that go into the small intestine to help break down biomolecules. So it is part of the digestive system. Yeah, pancreas does produce sodium bicarbonate. That is true. And Shalini, is epigenetics going to be on the exam? You should know that epigenetics and the tags that we talk about with epigenetics are like, yeah, like those are going to be on the test. You should expect those to pop up. You should know that that epigenetics are these tags are things that the environment can cause to shift, but you can pass them on to your offspring. So it's kind of like you can acquire a certain epigenetic setup in your life that you can pass on to your offspring. Okay, let's see. So those of you that picked the green one, shedding of the uterine lining monthly is not menopause. That would be menstruation. Menopause is the period of life when a woman stops ovulating for good. So that's more like when she's in her mid to late 40s. And that whoever picked this one, that can sometimes happen. The fetus could implant and start growing in the fallopian tubes, but if that happens, that's really bad news. That pregnancy will not last because the fallopian tube is not meant for a fetus to grow. Fetus should be growing in the uterus. Kesha, I'm not sure. I don't remember exactly what that was. Sorry, I don't remember the question and the answers. It might have been like phrasing, different phrasing. What is somatic cell gene therapy? Charveech, you answered that well. It does change your genes in your somatic cells, which is anything except your gametes. So like if you get gene therapy on your somatic cells, whatever disorder you have, let's say it's like you have some genetic disorder that's causing you some grief, or giving you some health issues. Somatic gene therapy would change that gene, or that gene that's causing that disorder for you. However, you could still pass that gene on to your offspring. So even though it's cured in you, or helped fixed in you, it's possible that you could give it to your child. Which is different than germline gene therapy, which would mean that you're actually trying to change the gene not just in your own body cells, but also in your sex cells, which is your sperm or egg, so that your child could not get it. So not only do you fix it for yourself, but future generations. Yeah, this one is just the idea that sexual orientation is not, you can't really tell who a person is attracted to sexually based off the fact that they are a transgender individual, because they could be attracted to any other gender. Good. All right, we got 15 questions left, coming down to the final. I'm going to probably call it a night after this. So if you have any questions about anything related to the final, get them in the next 15 questions. Good. Charvy, unfortunately, that's not an option. I can't just say, you can't take the final. You can just pass on the final. How are activators and tags different? I'm not sure about that one, actually. Like, I never heard the term activator. Let's see, what is an activator? I don't know. Is that something that Liz Lerner talked about? Like, is that something that would like activate a gene? Oh, okay. It's a transcription factor, essentially. Okay. Well, in that case, they're similar. So a transcription factor is something that will turn a gene on and off. And same thing with a tag. An epigenetic tag will also do that same idea. The difference is that an epigenetic tag is something that you can pass on to future generations. Whereas an activator or a transcription factor is not heritable. Does that make sense? Shalene, hopefully that makes sense. Like, one is heritable, the other is not. I think that's the key difference between an activator and a tag. Meenakshi, how hard would I rate the final? I would say the final is on par with the other tests in the class. Maybe just a little bit harder because it has more material. But yeah, I wouldn't say it's any much more extraordinarily harder than a normal test. It's just more stuff to know. Alright, that one you just gotta know the sequence. Stefan, if there is a blood clot in the brain, couldn't that also mean that someone is having a hemorrhagic stroke? A hemorrhagic stroke is one in which you have a rupture. So a blood vessel, an artery in the brain has burst, and you have internal bleeding in the brain. It's a hemorrhage. So that's not caused by a clot. A clot would cause a blockage of blood flow, and that's called an ischemic stroke. So there's two different types of strokes, ischemic and hemorrhagic. Ischemic is caused by a clot, hemorrhagic is caused by like an aneurysm, usually like a burst blood vessel. And so, I mean, it's maybe theoretically possible to have a clot, from a hemorrhagic stroke, but that's not the cause of the stroke. Let's just say that. All right. Remember, for this one, natural selection acts directly on phenotypes because it's the environment selects for the physical traits, the outward traits. So that's directly acting on like the physical trait, which is the phenotype. The genotype is the DNA that underlies it, that's in the genetics, that is the instructions for making the physical trait. Natural selection doesn't really act on that directly because that's not what's exposed to the environment. The physical trait is what's actually exposed. Danush, what are selection pressures? Can you give me 3 examples? Can you give me tree examples? You want some tree examples of selection pressures? So like, let's see, like one selection pressure. So a selection pressure is anything that causes an environmental, and a selection pressure is like an environmental factor or condition that causes different survival for different individuals based on how suited they are to handle that environmental factor, right? So let me give you an example. Like when we talk about the classic selection pressure, when we talked about, I don't know if you did the cormorant, the flightless birds case study, but when you think about the those birds, there was a selection pressure for shorter wings in that environment because, or in fact, there was a selection pressure against long wings. Really, is what it was. So like this, so like a long wing is beneficial for flying far distances, but on the island they didn't need to fly. There was no, the flying was not a factor. So the selection pressure on the island, in this case, was buoyancy, being able to swim in the water to get their food. And so larger wings were selected against because a bird wing is very, it's very light, not very dense, and it makes it harder to dive. It makes them float more. So it was selected against. So there was a selection pressure of catching fish that selected against individuals with long wings. And so over time, the wings got shorter and shorter because the ones with shorter wings had better survival rates because they got more food because they were less buoyant. They could dive and get food more. So the environment was selecting against long wings in that case, right? That's one example. Another example might be like there might be a selection pressure for some sort of camouflage situation, right? If you think about maybe, let's say, like a predator, like a lion, well, let's use like a what's a classic example? Like a tiger, right? The stripes on a tiger actually help it camouflage and be less detected. So there's the environment is selecting for tigers with better camouflage because they are less likely to be seen by their prey. They get a better chance of catching their prey. They get more food, are able to survive and reproduce and pass on those genes better. So they're selected for. I'm going to give you 2 examples. I think that's good. Oh, speak of the devil, here's a question that's perfect for this. Here's a selection pressure I was just talking about. I missed the question. Selene, okay. So are the tags in germ cells? Is it a memory thing? How does the environment affect things? Oh, Shalini, if she didn't, so here's the thing, if she didn't mention that, then that's not something you need to worry about, I don't think, for your test. But just to quell your freaking, if you're really freaking about it, just think of, that's all you really need to know is that epigenetic tags are these tags that can be placed on a gene or off of a gene, and they can turn a gene on or off. And they are in the germ cell, so they can be passed down from one generation to the next, right? So an individual can be born with certain tags already in place because their parents had those tags set up from their environmental interactions, right? So let's just say I'm like really stressed. Let's say I have a stressful life, and the stress causes my body to react to that. And one of the things is I need to now produce more stress hormones to deal with that. So my blood pressure goes up and all that kind of stuff. So the genes that produce those hormones, or those factors, the genes that are responsible for making that stress response, are going to be turned on. And that could be an epigenetic tag that I could pass on to my offspring. So they're born with that high stress epigenetic setup already established because of my environment in my life. Mr. Chang, you'd like my opinion on if I think every student should learn about basic trauma care and first aid? I think that's good. I think they should, yeah. I think that would be a really good thing. We teach it in physiology, but not everyone takes physio, so it's definitely not a required thing, but I think it would be good. You know, first aid is really important, CPR is important. I think mostly it's just more of like how to respond in a situation. I think most people need to learn how, like if some emergency situation goes down, whatever it is, the key thing is how do you respond? Like how do you not like panic? How do you avoid the bystander effect? How do you take action? Even if it's just calling, right? 911 or just checking on the victim. Even if you don't know how to do CPR, don't know how to do first aid. It's ideal if you knew how to do those things. But the key piece for me is like just how do you respond and keep a level head and, you know, either find somebody who can help or help yourself. Stefan, you do judo, nice. Judo, I don't know how you do judo. I do jujitsu. Judo, a lot of throws. Those throws are very painful. That's awesome. That's cool, dude. Cool. I like it. The biofinal is multiple choice. 100 multiple choice questions on a Scantron. Bring your number 2 pencils, sharpened and ready to go. Stefan, which STIs are curable, which aren't? Any viral STI is usually not curable, although some there are vaccines for, but any bacterial STI is curable. So like gonorrhea, chlamydia, curable. But things like herpes, HIV, those are non-curable. Yeah, and pubic lice is curable as well, because that's just like, it's like lice. It's like a parrot, like a small little insect. You can just use certain shampoos to get rid of it. What is genetic drift? Genetic drift is when the alleles in a population change their frequency over time, but just due to random chance, not due to any natural selection or environmental factor. So like, let's say that there's a population of humans, and there's no reason why, there's no advantage to having blue eyes over brown eyes, but just because of the random roll of the dice, blue eyes are passed on more often than brown eyes, and like the population shifts to having more blue-eyed individuals. That's genetic drift. It's just completely random. There's no advantage. It just happens to be random chance that's being passed on. And a polygenic trait is any trait that has more than one gene that's responsible for it. So, for example, like height, right? How tall you are is not determined. There's not one gene where it's like that's how tall you are. I got the 5'10" gene. Turns out that height is actually, there's several different genes that all contribute different amounts of height. So it's more than one gene. That's what that means. Nice, Kaushal. I like your explanation. That's good. And Stefan, yeah, gonorrhea is becoming antibiotic resistant. That is true. That is true. That's an issue. I'll have to look at that Instagram, Mr. Chang. North American Rescue. I'll take a peek. Aditya, yes, nutrition is on the final. You should know nutrition stuff. Glycemic index, fiber, healthy fats, that kind of stuff. Coming down to the last 2 questions. What is the difference between micro and macro? Just the scale. Like micro evolution is just like tiny, tiny changes, you know, within a population over a shorter period of time that might not be like super noticeable per se. Whereas macro evolution would be like long changes where you might actually end up with a whole new species, for example. Okay, the photos are graphic but educational. I'll be aware. Last question. Ooh, we've talked about this a lot. Are mono unsaturated or polyunsaturated fats worse? Which are worse? Don't worry about that, Stefan. Just know that both of those, the unsaturated fats, are typically more healthy than saturated for the most part. But the difference between the two, not a big deal. All right, how do we do? Yeet Yeaterson with first, Bot Time with second, Cool Cat 21 gets third. Nice. Good job, guys. Top 5, Charvy comes in 4th, and then November comes in 5th. Cool. So I'll just try and answer the last couple questions on here before I head out for the day. What else? Shalini, what is the difference between phytochemicals and antioxidants? Just the way I like to think about it is that like phytochemicals are a potent version of an antioxidant. So antioxidants are often like vitamins and minerals that are healthy for you, like to help with enzyme function in your body. And then phytochemicals happen to be these like potent versions of those that are found in like really vibrantly colored fruits. For the most part, those ones offer just slightly better benefits. So just think about, like, when I think about phytochemicals, I just think brightly colored fruits, like bright, thick berries or dark green vegetables. Yeah. Okay. All right. I see a lot of questions now all of a sudden popping up as I'm about to leave. I cannot answer all of them, unfortunately, at this point. Because, yeah, I gotta go. It's 9:15. I was planning on just doing an hour. It's been an hour and 15. So yeah, if you still have, hopefully people in the chat can help with the questions that people popped up in there. Danush, you had a lot of questions there at the end, so hopefully people can help you out with those. But if not, a lot of those questions you can look up online, you know, so I wouldn't worry too much about it. But I appreciate y'all tuning in, tuning in. I will see you guys on, what is it, on Wednesday, right? Yeah, Wednesday. All right. Peace. I will see you on Wednesday. Good luck with those of you that I will not see. I hope you do well in your finals. I hope your week goes well. And I hope you all have a great summer. and enjoy your summer break, because that's what it's all about. That is what it's all about. Okay. Bye, everybody. Have a good night.